All right, let's get started with today's webinar. I'm Rachel Hostetler, and I'll be moderating this webinar today on single-cell and single-nucleus transcriptomics with Matthew Schmitz. And as a reminder, this webinar is being recorded and it will be posted online to our YouTube channel afterwards. And this is part of a webinar series that we're doing once a month. So if you'd like to register for any of our future webinars, please do so. We are also taking questions during this webinar. If you have any questions, please put them in the chat and I will read them aloud. And if you're watching a recording of this later on YouTube, if you have questions then, please post them on our community forum, which is at community.brain-map.org. And it's also written in our description on YouTube. And so we're scientists from the Allen Institute for Brain Science, and there are some common goals and principles that we have at the Allen Institute in the way that the Allen Institute runs. So the Allen Institute is a nonprofit bioscience research center. And one of the ways that the Institute runs is based on team science. So we have these very large interdisciplinary teams of groups doing very specific parts of a research pipeline, which leads us to these really large, big science projects, which are very large scale with a lot of robust and massive amounts of data. And we also believe in open science. So we share our research publicly online for others to use on brain-map.org. And so today's webinar specifically is focused on single cell and single nucleus transcriptomics and how we use it specifically at the Allen Institute. And here are just a few ways that we use it in some of our projects. Creating cell type taxonomies, figuring out, what is the parts list of the brain. Observing how cell types change over time, perhaps with age. Observing how cell types change during a disease progression. Observing how cell types compare across species in evolution. Observing how cell types are generated and how they might change during development. And also for the genetic tools that we create, using transcriptomics and RNA sequencing to analyze are our genetic tools specific to a specific cell type. And there are many more examples where we use this technology here at the Institute, but here are just some broad examples. And regarding the parts list of the brain, well, in 2023, a whole mouse brain taxonomy was published using single-cell transcriptomics and also spatial transcriptomics to map out that parts list of the mammalian brain. And what that ended up looking like was a very large taxonomy of cells, starting at the top with 34 different classes of cells. And then within those 34 classes, we had 338 subclasses, then 1,201 supertype. And then at the very bottom of our taxonomy, at the finest level, 5,322 clusters were identified. And this was all done using single-cell RNA sequencing. And if you'd like to view this taxonomy and learn more about it, you can go to brain-map.org and you can view this taxonomy and interact with it in our online tool called the ABC Atlas, which stands for Allen Brain Cell Atlas. Another example of a recent project using single-nucleus RNA sequencing and seeing how cell types change with disease is our human Alzheimer's taxonomy through the CAD project, or the Seattle Alzheimer's Disease Consortium. And so in this taxonomy, there was a range of severity of disease progression within the cohort from the donors. So some had very severe states of disease, some had high levels of dementia, some had no cognitive impairment at all. And by doing this analysis and combining all these different members of the cohort through the transcriptomic data, the researchers here were able to see which cell types were especially vulnerable in disease or were present in disease that were not present in non-disease states. And we can see that here in our glial cells, marked over here by this red arrow. These cell types here have CAD written in their description here, which means that these were microglia that were specific to being in an Alzheimer's state. And so by doing transcriptomics, by creating these taxonomies, this is just one example of how we use this technology at the Institute and how it's used to look at how cell types change or appear or are different throughout disease progression. And we have an entire webinar focused just on this project here, our Alzheimer's Disease Atlas, next month on February 12th. So make sure to register for that webinar if you're interested in hearing more about this data. And now I'm going to hand it off to Matthew, where he can go into more details about how RNA sequencing is used at the Institute.
Great. Thanks, Rachel. Yeah, that's a great introduction to yeah, how the Institute's using single cell. And yeah, so I'll tell everyone a bit more about, I don't know, the kind of overarching how and why and what do you do to make it happen when it comes to single cell transcriptomics. So yeah, just to give everyone an idea of who I am and why I might be qualified to talk about this. So I started my career actually with some fluidigm single cell RNA-seq during my time as a sequencing core technician at the University of Wisconsin-Madison. So it's been almost 10 years of single cell in my life. I then did my PhD at UCSF with Alex Pollan and Jimmy Yee, using single cell RNA-seq to study brain development. And yeah, so we studied inhibitory neurons across species and found some fun things. And so now I'm currently working with Trygve Bakken as a computational scientist on the EvoGen team here. And so as part of this work, I'm using single cell multiomics, RNA plus attack, and then also spatial transcriptomics to study the differences across 15 different mammalian species in their primary visual cortex. And on the side of that, I'm also building a cetacean brain atlas. And so, yeah, so the roadmap for this talk, I will attempt to give you all a molecular basis for single-cell RNA-seq, which includes the questions that it's meant to answer, the type of samples that you put into it, and then the libraries that you generate, these pieces of DNA with all sorts of information encoded into them. And then next, really giving sort of the foundations for the analysis, how you get from these molecules to the data that you actually analyze, and then just the broad techniques that you use to analyze it, and then sort of look at the future and the broader landscape of omics. Yeah, and so this is an intro. I aim to get you sort of to the starting line for generating and analyzing your own single-cell RNA-seq experiments. If you're already an expert in them, maybe you'll learn something, maybe not. But because it's so broad, I won't go too deeply into any one thing. Yeah. Okay, so yeah, just to get started, I figure it's worth stating sort of the goal of single-cell RNA-seq. And to me, this is, I mean, pretty obvious. The goal is to read out mRNA transcripts in such a way that you know how many there were to begin with in their cells of origin, and then to be able to know which cells that each transcript came from. And so then you can use these transcriptomic profiles where you have a profile for each cell to understand a bunch of things. And some example questions are looking at, say, the heterogeneity of a population of cells. How many different cell types are there? Or are two different populations of cells composed of the same types, or are there types that are there in different abundances? You can also answer sort of your classical RNA-seq differential expression questions, but at a single cell level, and looking specifically at a cell type, which is a major struggle of sort of bulk RNA-seq. If you have a heterogeneous population, your differentially expressed genes tend to be driven by population instead of like actual changes within each cell type. And so yeah, so the question is sort of, how does a given cell type differ across species, as an example? That's the question I ask a lot. And lastly, you can get towards more complex things like expression dynamics, and say you have a developmental trajectory where cells are differentiating. You can ask questions about what genes are changing along this trajectory. Yeah, and sort of a final note, but foundational note, is the single cell versus single nucleus RNA-seq. And to me, these are essentially equivalent. And through this talk, I'll basically use them interchangeably. The difference is obviously that single cell uses whole cells and sequences all of the RNA in them. Single nucleus just sequences the nuclei. And so, yeah, because of this, you get some differences in the data and sort of the limitations of them. Specifically, for single cell RNA-seq, you can only use fresh cells because the whole cytoplasmic membrane doesn't survive, say, flash freezing. Whereas for single nuclei RNA-seq, this is sort of revolutionary because it's able to be used on flash frozen tissue that's been stored at -80, and sometimes for 20 years, because people have been banking tissue for a long time. And so that's really the power of it. And so, yeah, you capture more mature transcripts and isoforms using single cell RNA-seq, obviously because you're including the cytoplasm. And so, yeah, so sort of on the bottom, you can see these UMIs and genes, the number of counts of each of those that you get per cell. And you can see that in whole cells versus nuclei, you just get more transcripts that you read out. But to balance that out, single nuclear RNA-seq is kind of more flexible, because in addition to using it on frozen tissue, it also is better for capturing hard to dissociate cells like vascular cells or very large neurons. And along with that, the sort of composition that you read out is more reflective of the original composition of your tissue because you have fewer dissociation artifacts. All right, so that's the difference between those, scRNA-seq, snRNA-seq. I'll just use them interchangeably, but yeah, it'll depend on what you're doing and what your goals are for which one that you choose. Okay, so to get started, I figured I'd just sort of walk you through how this data is generated at the Allen specifically, and how we get from tissue to yeah, this data. So it all starts with some frozen tissue, where here we have a brain slice. And so, yeah, so in order to get started, you pretty much can just chop a piece off of your tissue. You have to warm it up to -20, but yeah, in the lab, we just use a razor blade and you cut off your region of interest in the right amount. And then you're ready to do a nuclei prep. And so, yeah, so once you start with your tissue, here you can see this Ramoni-Cajal drawing, and see how tangled up all these neurons are. You have to sort of get them apart. And so if it's whole cell, you have to dissociate them in a gentle way so that they don't all explode. But for nuclei, you can take this frozen tissue and do sort of a Dounce homogenization or other methods to isolate your nuclei. and to clean them up and to remove myelin and stuff like that. And so what you get is sort of a mix of nuclei, and even some whole cells can survive this process. And that's why the next step, which is FACS or FANS, is very important. Well, for multiple reasons. In a lot of our experiments, we do this sort of regime where we use nu-N stain and try to capture 90% nu-N positive and 10% nu-N negative, because the diversity of neurons in the brain is just higher. And so we want to spend more money on that. Yeah, so in doing that, you also get rid of some of the junk. You capture the cells you want to capture. Yeah, and that's sort of how you get to the stage where it's ready for the actual single cell sequencing part, because you can either flow sort right into a plate for experiments like SmartSeq or split pool based single nuclei transcriptomics protocol, or sort of your classical droplet based transcriptomics, which I'll spend more time on here because I would say the vast majority of transcriptomics that we do at the Institute are 10x genomics, sort of the most common type of single cell transcriptomics. Yeah, so basically you take your mixture of cells, you mix it with some reagents, including reverse transcriptase, and you put it through a microfluidics channel, which mixes in these gel beads with your cells, and they end up in this nice little well, and that's within this nice little microwave machine that you put the fun chip into. Yeah, and so what you get out is an emulsion of oil and these droplets of water, reagents, cells, and beads. And so the first step, once these cells are in their drops, is that you sort of lyse to release the mRNAs here with their little poly A tails, so that they can leave the cell. And so in drops with a bead, sort of what these beads are is this like dense lawn of these DNA sort of probe sequences, which come with a bunch of sort of information sort of added onto it already. So on each gel bead, there are these pieces of DNA. They have an adapter so that you can do PCR. They have every sequence on the bead has the same cell barcode. And that's important because then you'll know which droplet things came from. Then every piece of DNA on the bead has a different UMI, a unique molecular identifier. And so then you can know which original molecule things came from when you go back and sequence. And then finally, you have some sort of capture sequence. And this doesn't have to be poly-T, but for 3' single-cell RNA-seq, you're capturing the poly A tails on mRNAs. And so yeah, so what happens is that the poly A tails just anneal through sort of your classical Watson-Crick base pairing to these pieces of DNA. And this is able to act as a primer for reverse transcriptase. And so then you're able to sort of sequence your first strand of complementary DNA or cDNA. And yeah, from there, it's little molecular magic to make your second strand of DNA and do a little bit of amplification. But sort of the first thing that you get out of these library preps is this piece of DNA, which has an adapter on one end, a cell barcode, the UMI, the poly A, and then sort of your transcript sequence. And then this is a template switch oligo, which you're not going to end up with because these are very long pieces of DNA. They include the full, or as long as the reverse transcriptase could reverse transcribe. And so, yeah, the thing to know about this intermediate step is that only one end has the cell barcode. And you can't put something this long into, well, you can't PCR amplify it efficiently. And also for sequencing, you need small pieces of DNA for technical reasons. And so to get to your final library, there's this tagmentation step where you basically stick a adapter into the middle of your transcript sequence. And so then you get this nice short piece of DNA with adapters on each side where you can do PCR, or there are also the read primers for Illumina sequencing, coincidentally. And yeah, so you have this library that has all this information plus the transcripts encoded in the DNA. Yeah, and so, right, some notes on this. The fragmented transcripts that you get out, it's about 200 to 500 base pairs of sequence at the 3' end of each transcript, or you can also capture the 5' end or specific capture sequences. But if you look at sort of this read pileup of sort of where your reads end up, if you have this gene annotation where the 3' end of the gene is sort of on the right, you'll see that for SmartSeq, which we'll talk about in a second, and TrueSeq, which is bulk RNA-seq, you get reads along the whole length of the transcript, whereas for 10x, the reads are all within like 500 base pairs of this end of the transcript. And this means that you don't read out the whole thing. But on the other hand, every transcript only has one poly A, so it's easier to count how many transcripts were in a cell if you only capture the poly A and only sequence the poly A linked reads. Yeah, and so you end up amplifying this with PCR, but the UMIs on these transcripts mean that you can actually count how many original transcripts there were in the cell, which is sort of unlike old school RNA-seq methods, where you have PCR bias, and that's just a thing you deal with. Yeah, and so then you take this library and you're going to read it out using sequencing. But before we get to that, I just wanted to give a quick shout out to some of the plate-based methods, which work differently because they partition cells into each plate of, say, a 96-well plate. But they work very analogously in that you're doing reverse transcription, and then within your little partition, you're adding a cell barcode, and you get pretty similar type libraries out of this, where you're reverse transcribing, barcoding, fragmenting, and amplifying. And so what's the point of these different platforms? So droplet based is great because it's high throughput. You can get hundreds of thousands of cells in a single experiment. You get pretty high quality transcriptome capture, and you can capture cells or nuclei. Now this plate-based sort of SmartSeq 3 style of single cell RNA-seq, it has its own strengths. It's very low throughput because you have to put a cell in every well. So a few hundred, a few thousand cells is really as much as is reasonable, because you have the library prep in those plates, which gets to be a lot of work. you get kind of the highest quality transcriptome capture, like you get many more transcripts per cell using this method. You also get full transcript length coverage, which means you can see which sort of exons are used or not used. You don't quite get the full isoform reads because you need long read for that. But yeah, and based on the capture, this is also a method that's compatible with, say, patch-seq, where you do some electrophysiology on a neuron, then you suck out the nucleus with your pipette, and you just stick it right in the plate. And so, yeah, it's great for stuff like that. And yeah, it can also capture cells or nuclei. Lastly, I just wanted to highlight this sort of third school of methods because they're very high throughput, and that's this split pool index single cell RNA-seq, which is sort of the Sky RNA-seq family. And so yeah, you can do millions of cells per experiment in this. It's great because you can multiplex different biological conditions without doing anything special. It's quite cheap when scaled, but in my experience, you get sort of the fewest reads per cell and sort of the lowest quality of transcriptomes. But yeah, and it's compatible with other omics along with that, which we can talk about later as well. And I just wanted to highlight this resource now that I've sort of given you the broad strokes of these libraries, because as someone who had to do all the steps of this during graduate school, yeah, so Sarah Teichmann's group keeps this GitHub repo with all the library prep sequences, sort of from the beginning through each step. And then in sort of plain text, you get the actual DNA sequence. So you can create your own primers. You can figure out sort of how you want the sequencing core or whoever to do your sequencing. And this was a lifesaver, honestly, to have this sort of open information. And so I just wanted to highlight it for people who are trying to figure out what's going on at each step. Yeah, another thing that I wanted to just give sort of a broad strokes on, like the cost of single cell RNA-seq is going to depend greatly on like what reagent kit, like where you are, how deeply you sequence. Basically, it can vary widely, but just sort of a conservative estimate is about 30 cents per cell for an experiment with 20,000 cells, which would end up costing somewhere in the neighborhood of $6,000. However, prices are falling. There are ways to make this cheaper by, say, multiplexing different individuals such that you can run more cells per lane. But yeah, anyway, maybe that is useful to you at some point as you plan experiments. Right, so I've explained to you how these libraries are generated. Now, how do we actually get from these libraries to a count matrix, which is really sort of the starting point for analysis? And the way that you do that is through your sort of classic sequencing by synthesis, which is like Illumina sequencing in the US. So yeah, so we just walked through the library generation or library preparation. Then basically these sequences go onto these glass flow cells where the pieces of DNA kind of bind and amplify, and then you sort of add these colored bases and you sequence the sort of the transcripts again, one base at a time, and you take pictures of this flow cell, and whatever color it is what base there is at that position on that transcript. And and most of the time you don't need to know how that works. You just send it off and you get your data back. But I guess I would be remiss if I didn't mention that there is also sort of a third generation of sequencing, which is not usually used for single cell genomics. Firstly, a few years ago, DNA nanoball sequencing came out, which is really a second gen method, but is new, so it kind of gets a 2.5th gen label. But it's basically a slightly cheaper version of Illumina sequencing. And sometimes it's not even cheaper, but just in case you hear or see that, it's essentially you get the same read lengths as Illumina sequencing. And so, yeah, so as opposed to that, the true third generation sequencing are these long read sequencing methods. And sort of the main methods in this space are Oxford nanopore sequencing, which can generate these extremely long reads, but they have the shortcoming of having a lot of errors in the actual bases they read out. But you can get reads that are greater than 100 kilobases. And so you can easily read out transcripts from their poly-A-TL to their 5' cap, and see all of the exons included in every single molecule. And then lastly, there's PacBio sequencing, which also has long reads and has a lower error rate. And so, yeah, generally these are used for sequencing genomes more than transcriptomes, but they can be useful if you're interested in isoforms. All right, so once you have your sequencing data, you're going to start with your reads. Then you map them to the genome, and you have annotated transcripts because people know where transcription happens and where different isoforms come from in the genome. And so, yeah, you map them to this or a K-mer database for the non-alignment based methods. And I will say for cross-species work, this is where you need a different species genome with a different transcript annotation. I mean, you always need to pick your right species, but this is just one of the places where it becomes challenging to do cross-species work. Generally, these pipelines then clean up your cell barcodes, and then they're going to count the UMIs for each gene. And so just a couple popular methods. There are the alignment-based methods, which are a little bit slower. These include 10x's own Cell Ranger or Star Solo. And then sort of in opposition to that, there are these non-alignment-based methods, which quantify transcripts. And that could be Callisto or Alvin, Fry type things. And so, yeah, so what actually ends up happening here, what you get back from the sequences, the sequencing core would be these FASTQ files, and this contains sort of an ID for every read, the sequence of every read, and then sort of base qualities. And then, yeah, you give this to, say, Cell Ranger or the Star Aligner. And you get these BAM files where it takes every read and it maps it to a location in the genome. And you can annotate what transcript that location is inside of, or if it's not within, then it's maybe junk or it won't get counted. And so, yeah, so this flags attributes, it gives the sequence, it gives the quality of the alignment, and the location. And so given all these reads that are mapped to the genome, you can then quantify them to turn into a matrix that you're interested in. And sort of the most basic file format is, well, the most basic would be a full matrix. But for these sparse matrices, which I'll cover why they're sparse in a second, basically you count anything that's not zero. And so you get these sort of row, column, and then count matrices, these .mtx files. And then, yeah, so you can put this into a container file. And my favorite is and data. There are a few, like R has its own. I'm a Python person, so I ride for ScanPy. Um, and so, yeah, so what these file formats are designed for is containing many types of data all together in this database. They One of their great strengths is that they can integrate this sort of out of memory data processing, um, where most of the data stays on the disk, except you can load some of it into memory as you need to. And so really the core of this is that sparse matrix I was just talking about, this X. And then sort of on your dimension of cells, you can have metadata or dimension reductions where you have these vectors attached to each cell. And they form these big matrices. Or you can also have those for genes, um, where you have metadata, which would go in this bar, or you have like a big correlation matrix of all the genes. This goes in this bar P. And then you can put miscellaneous things in this file. But you will almost certainly see these files doing single cell RNA-seq analysis. Yeah, which I hope to give you a small foundation in here. So yeah, so what are we doing with the fundamental analysis? Um, so just for demonstration purposes, um, I've taken an example brain data set. And here we just have sort of the cells and the genes, and the color is representative of the log 10 counts per gene. So everything that's white here is a 0, and everything that's not is some number of counts that's greater than 0. And so, yeah, just to sort of give you what these data sets are composed of, 83% of the values in this data set are zeros. So it's very sparse in that way. And yeah, so in addition to that, a lot of the transcriptome counts end up going to a few genes, where the top 100 genes get 29% of the counts. If you expand that to 2,500, you get 76% of the counts. And so, yeah, and it's important to note that these are not all the transcripts that were in the cell. In a perfect world, we would read them all. But in reality, you just get this snapshot of something like 1% of the mRNA, and it varies by cell type, but within an order of magnitude or so of 1% of the mRNAs in a cell. Yeah, so once you have your data, usually the first step is to do your quality control. And what you want to do is remove bad cells or bad droplets. And there are a number of things that can make a cell bad. And this is just noise that is going to make finding cell types and doing differential expression more difficult. And so you want to get rid of drops with low UMIs. You want to get drops that have doublets, which are multiple cells end up in the same drop. You want to get rid of debris. You want to get rid of dying cells, because this can save you a lot of time further on in your analysis. A helpful tip that, just from my experience, that I thought I'd share is that bad cells tend to kind of pull together into their own clusters. And so sometimes you could wait to do your filtering and do sort of an initial clustering, and everything that's junk will end up in junk clusters, and then you can just get rid of them from there. Or you can just filter them by setting thresholds. And I don't know, maybe it's controversial, but I say when in doubt, kick it out when it comes to sort of RNA-seq questionable droplets, because it'll create big headaches and possibly retractions later on if you think things are cell types, but they're actually just junk. But you have to be careful because sometimes things are legitimate cell types that you just don't recognize. So be very careful in what you throw out. Anyway, so yeah, as I mentioned, poor quality control can undermine what you're trying to do here. Yeah, so what you start out with is this expression matrix, which is the size, the number of cells times the number of genes. So for a big data set, this could be 1 million cells times 20,000 genes, which is an enormous matrix. And it's too large for doing, well, all sorts of different algorithms, both in terms of computer memory and computer processing. And so dimension reduction is really a foundational method of single-cell RNA-seq. And so the point of dimension reduction is to decrease the dimensionality in the data while maintaining the essential attributes or relationships between your data points. And so, yeah, you need this because high-memory compute nodes are expensive. And you can see here, this is sort of a representation where you start out with a 3-dimensional data set. You could imagine it being a 20,000 dimensional data set. And you project it down to a lower number in a way where the cells that are more similar to each other stay closer to each other. And then you can even make it linear so that you could draw a cutoff on this line. Yeah, and so one of the most common types of dimension reductions that you'll see is a little bit down the line. But because you see it everywhere, I figure it's the one I should cover first. And that is this idea of a UMAP or a t-SNE. It's these visualization dimension reductions where the idea is to collapse all of your dimensions down into just 2 so that it can be easily visualized. And they should only be used for visualization. But the point of these is that points that are close to each other in this 2D space represent cells that are the most similar in higher dimensions. So you can see each type sort of pulls together in its own area of this UMAP plot. And yeah, that kind of demonstrates what a UMAP is all about. Yeah, so in order to make a UMAP, oftentimes the first step is to do sort of these linear matrix decomposition methods. Principal component analysis, or PCA, is kind of the most common one of them, but there are also like independent component analysis, latent Dirichlet allocation, non-negative matrix factorization. All of these basically do the same thing, which is decomposing X into a matrix that is the number of cells by some number of new components that you're creating. And so this is like your cells in principal component space. And then genes by components, which is like your gene loadings onto the components. And basically multiplying these together reconstructs your original matrix is the idea. But the dimensionality of this, say, W matrix is much smaller than your original V matrix. So similar to this, but also different, is doing dimension reduction based on deep learning. And so these are variational in autoencoder methods. SEVI is the most prominent one for single cell. And we use this a lot for cross-species work because they're able to do this nonlinear transformation to put all your cells into a latent space. And then you can also sort of, as part of the middle of this thing where you have a black box, which transforms your original data, and then you have latent space, and then some other covariates, and then you have another black box, and then you reconstruct it. Basically, you can co-embed and remove certain effects from the data by letting these neural networks account for, say, the gene expression differences that they drive separately from the latent space. And this is a little complicated, but I think if you just look at these UMaps, it's a good demonstration where you can see that the red and blue, these are nuclei data sets, and then these are cell data sets. And so these tend to be, they have differences because there's cytoplasmic transcripts in the cells. But by giving SCVI the sort of data set that they were generated in, it's able to account for these differences and put the cell types that are the same, regardless of what data set they come from, into the same area of the latent space. And so you can cluster them together. And so, yeah, so that's really the next step. Once you have a dimensionality reduction, it's typical to do some sort of clustering, and the most popular method are these sort of graph-based clusterings, where you take each cell, you find the cells that are nearest to it, and you make this graph. And then there are methods like Louvain or Leiden clustering, which can detect these communities in the graph, or just in our parlance, detect cell types in your data set. Yeah, and so at the Allen, we use sort of a modified version of Louvain, which is this Scratch HiCat, where there's a Python implementation as well. And so it's basically iteratively doing Louvain clustering, where you cluster and you take each cluster and you cluster that. And then you do a differential expression between the clusters at each level, and you merge the ones that don't have significant differentially expressed genes. Yeah, and so basically at the end of the day, you get the smallest clusters that have gene markers. I guess is the simplest way to think about it. But there are a ton of clustering methods, and they're useful for different purposes. Okay, and then, yeah, maybe the most classical question and type of analysis for RNA-seq in general is differential expression. And this is basically asking, given conditions and a cell type, which genes are significantly different between the experimental condition. And kind of embarrassingly, in the field, the best practice isn't to look at all the cells as data points and to sort of hierarchically manage the variance of biology and technical. Basically, pseudobulk methods are still the most robust, where you say take the mean of each replicate for each gene, and then use sort of these old school differential expression methods on your biological replicates. And so these are methods like DESeq2 or Limma or EdgeR, which are basically standard generalized linear models. And there are other ways to do differential expression, for instance, SCVI, which I mentioned a second ago. It can let you do some DE in sort of heavily batch affected data. Okay, so yeah, so just a quick run through of the many methods, and I'm not going to actually list out the methods, but basically there's quality control. Basically, you're demultiplexing, removing doublets, removing junk cells. Batch correction methods, which is, I guess, self-explanatory, hopefully. There are pseudotime methods, which say you have a developmental trajectory. You can order the cells along it from, say, earliest to latest in development, that type of thing. RNA velocity, which sometimes points you in the exact opposite direction. So you can take that as it is. Differential composition testing, which are statistical models, which are meant to answer, say, is this brain region made of the same cells in a human and a chimpanzee, or are they different in composition? You can look at co-expression networks to see sort of which genes are most correlated with each other, which gets you towards like regulatory networks. And sort of similarly, there are databases of receptors and ligands, which can allow you to start to infer which cells might be talking to which other cells that are listening. There are methods to impute counts to make your data set sort of less sparse. I don't endorse that. It exists. And finally, there are a ton of miscellaneous methods for different questions you might have depending on your field and your system. These are like T cell receptor sequence analysis, lineage tracing, finding structural variants, etc. Yeah, so lastly, I just wanted to point out that there are a lot of methods that are sort of used in tandem with single-cell RNA-seq to answer different biological questions. My favorite right now is spatial transcriptomics. I mean, it was super hot in 2023 and 2024. Everyone had a spatial method. And so this basically fills the need that dissociating your cells explodes your tissue and you don't know where your cells came from. Spatial transcriptomics either sequences on a slide or you use like fluorescence in situ hybridization probes, many of them. And so you're able to look at the location of different transcriptomic features, but you get fewer genes and less transcriptome death than, say, DropSeq. And so I think it's best used together with single-cell RNA-seq, where you get a high-resolution transcriptome and you can detect all the types, and then you design a gene panel, basically, or do a sequencing thing. And this allows you to sort of figure out where different cell types come from and what their spatial distribution is. Yeah, and so it's 2025. Basically, you can combine mRNA with any type of genomics you can imagine. This is a non-exhaustive list of the combinations that exist. And yeah, so if you can imagine it, you can do it and answer those questions. And speaking of questions, I think I am ready to take any questions, or yeah, we can wrap up.
All right. Thank you, Matthew. We do have a really quick survey that if everyone could fill that out, that would be great. And this survey is just here. It's anonymous and it helps us plan our future webinars since this is part of a webinar series. So it'd be very helpful. It should just pop up on your screen here. And so if we just take a few minutes, if people could fill that out. Again, it's anonymous. This is just helpful for our future webinars and things we would like to plan and improve them for our audience. I'll leave that up for another minute or two. And it looks like while we have that survey going, we do have some questions here. So the first question is related to that very last slide that you showed, Matthew, that has all the different ohms on there. And someone's asking about, is there, a paper or a citation related to that particular slide?
Yeah, it's this Soy et al. 2023 paper. I think it comes up if you Google like single cell multiomic.
B-A-Y-S, yeah, B-A-Y-S-O-Y is the last name, Basoy. I can also type that in here, Basoy et al. 2023. All right. And then another question that we have is, can you talk a bit more about the processing of large data sets, such as best practices?
Yeah, I mean that it's kind of the trick of single cell. So I mean, best practices are, well, it depends if it's best practice related to like computational resources, or yeah, I guess it would need to be a more specific question. I could say that like deep learning handles this question in a way that say you keep the entire data set out of your memory, and then you load little batches of cells at a time. But by randomly going through your whole data set, you're able to work with giant data sets, but never have to sort of load them all at the same time. I will also say that for very large data sets, oftentimes subsampling them can get you started. I think that's a good practice. Um, in general, the bigger your data set, the more sensitivity you'll have to detect rare cell types. But don't discount sort of FACS is one example, but there are many ways to enrich for cell types that you're interested in. And you can save a lot of money by actually using the biology to select for what you're looking for. And so you don't need as big of a data set in order to see all the cell types that you're looking for.
Right, I think that's a pretty good answer. We only have a minute or two left, and we do have a couple more questions. The first question is there a need for a heavy computer science background to carry out such computational methods?
I would say no. So the field is mature enough at this point that there's sort of a beaten track where you can do all of these basic steps, just sort of copy pasting code from tutorials. And so you don't really need a comp bio background for that. And then there's an off-road track where you're sort of developing your own methods and sort of tearing things apart and putting them together. And that's where like understanding the algorithms and like the files and all sorts of those things, there are many different realms of knowledge that you have to collide in order to sort of go off road. I think that's where the fun is, but yeah,
Yeah,
to get answers you don't necessarily need.
I was going to say, I think that's where things like GitHub really come in handy, because you can see what other people are doing, how they're doing it, whether or not that fits with your experiments and your design. Just people, openly sharing code and how they do things is very helpful in that regard.
Exactly.
Another quick
The way I think about it is R is less good as a programming language and better as like you write one command and it does a whole bunch of things. Whereas Python is more of a programming language and you might have to write more lines of code and it's more flexible And it's more integrated with deep learning and stuff. If you already know R, I would do R. If you already know Python, I would do Python. If you want to do deep learning, I would do Python. Yeah, I don't know. If you work in a lab that everyone is doing R, I would do R.
We definitely have people who use both at the Institute. Again, if you go to the Allen Institute GitHub, you will find, yeah, you'll definitely find certain things that use Seurat and use R, and other things that use Python. So it seems, yeah. But
Yeah, you
which
have to
language
look at
you're
your own
comfortable with?
situation, I think. Yeah,
Yeah.
exactly.
I've seen people, yeah, use both for many years. Okay. And we have about one more, I'll answer one more question since we are a little bit over time. So regarding imputation, are the imputation methods accurate in imputing missing values and addressing the sparsity problem?
I guess the answer is, well, yes and no. I mean, there's not really a good way to test them, is kind of the problem. Um, there are certain cases where, I don't know, I think imputation is complicated and is best avoided unless you really know that it's necessary. Um, the risk is that you make yourself more confident and you believe you have more data than you actually have. But I mean, yeah, yes, they are accurate in imputing missing values, but they don't add information, I guess, is what I'll say. Um, like you're just kind of strengthening the means of your clusters, if that makes sense, and not necessarily. Yeah, I don't know. Imputation is complicated.
Yeah, unfortunately, I don't have more time to talk about that particular question in depth. But I do want to mention that again, we are part of a webinar series here. So we have a few more webinars coming up. You can register for all of them at alleninstitute.org/events or click on this QR code. And also community forum questions. If you would like to continue this question of, imputation, you can post that on communityforum.com. Maybe there's already a thread talking about this particular question. I would highly recommend going to our community forum where you can interact with more scientists, hear lots of different perspectives from people in different parts of the Institute, and also look at questions that have already been previously answered by some of our scientific staff. So definitely check out this community forum for some more technical questions. All right, thank you everyone for joining us today, and thank you, Matthew, for your presentation. Have a nice